Hello, am I audible?
Yes, Aditya.
Yeah, okay. We should start now. A very good afternoon to everyone. On behalf of Junior Science Club and Entropy, I welcome you all to Nobelium 2020, a webinar series based on the Nobel Prizes given this year. This talk is about the Nobel Prize awarded to medicine or physiology for the discovery of hepatitis C virus, which explained a major part of blood-borne hepatitis cases. The Nobel Prize was awarded jointly to Harvey J. Alter, Michael Houghton, and Charles M. Rice. The discovery of hepatitis C virus revealed the cause of the remaining cases of chronic hepatitis, apart from hepatitis A and B, and made possible blood tests and new medicines that have saved millions of lives. I am glad to introduce the speaker for this talk, Professor Sharmishtha Banerjee. Professor Sharmishtha Banerjee received her PhD in biochemistry in 2005 from the University of Hyderabad. She worked at Center for DNA Fingerprinting and Diagnostics, Hyderabad, India, and later continued her postdoctoral studies at the Laboratory of Transcriptional Biology on mechanisms of road-dependent termination in E. coli. She worked briefly at the Molecular Cardiology Unit, Lerner Research Institute, the Cleveland Clinic, Cleveland, USA, on the role of integrins in prostate cancer metastasis. Presently, as a professor in the Department of Biochemistry, School of Life Sciences at University of Hyderabad, she teaches immunology and enzymology along with various topics on infectious diseases and host pathogen interactions. The research focus of her laboratory is molecular pathogenesis and immunology of Mycobacterium tuberculosis (MTB), human immunodeficiency virus (HIV), and MTB HIV co-infection. Her honors and awards include Professor B.K. Bachawat International Travel Grant for Young Scientists 2018, Chancellor's Award 2017, University of Hyderabad, National Women Bioscientist Award Young Category 2016 from Department of Biotechnology, Government of India, Innovative Young Biotechnologist Award IYBA 2011 from the Department of Biotechnology, Government of India, BioAsia Innovation Award 2008, and INSA Young Scientist Award 2005 from the Indian National Science Academy. With this, I now invite Professor Sharmishtha Banerjee to start the talk. I request the audience to put their questions in the chat box of Google Meet or the YouTube live stream. Over to you, ma'am.
Ma'am, you're muted if you don't mind.
Ah, okay. So thank you so much for introducing me to the audience. And I begin with thanking the organizers, Entropy Club and Junior Science Club, for starting this series, which very selfishly is very close to my heart as well, because I've always been trying to understand about the Nobel laureates, especially in other fields. And your series has allowed or a vast audience to understand why these eminent people have received this Nobel Prize, what is the work they have done, what is their contribution, and I thank you for that, and thank you for making me a part of it. So I'll start. I'll share my slides now so that we can start the discussion. Let me know if it is visible. Is it visible?
Yes, ma'am.
And I'm audible too.
Yes, ma'am.
Right. So I'll talk about the Nobel Prize in Physiology or Medicine that they received, they gave in 2020. The Nobel Prize in Physiology and Medicine was given to Harvey Alter, Michael Houghton, and Charles Rice for their discovery of hepatitis C. So even before we understand the journey that they had taken, to appreciate the work they have done, let us first quickly understand what it means to the society, this discovery of hepatitis C. Liver, as we all know, is a very important organ, especially for invertebrates, and it performs many life-sustaining functions. It participates in detoxification of various metabolites. It synthesizes various kind of biochemical molecules, proteins, which are important for sustaining growth, for digestion. Its role in metabolism, in regulation of glycogen storage, decomposition of various red blood cells, etc., makes it a very, very important organ. Once in a while, because of several kind of conditions, the liver gets affected, and there is a situation which makes the liver inflamed. And this inflamed situation can become slowly worse, and you see a kind of scarring of liver tissue, which we call cirrhosis. And over a period of time, if it is not checked, it leads to a cancer-like situation of liver, which we call as hepatocellular carcinoma. So this condition is called the inflamed liver or hepatitis. It's an inflammation of the liver which destroys the liver and definitely takes millions of lives if such kind of condition occur. Now, the liver hepatitis can be because of certain infectious agents, like it can be viral, bacterial, fungal, or parasites, but also the damage can be because of certain non-infectious agents, like sometimes drug abuse, excess of alcohol can be case of autoimmunity as well as certain metabolic disorders. We'll not talk about the non-infectious causes, but in the case of infectious causes, there are various ways that this infection can happen in our body. That can happen through contaminated food, water, or sometimes because of substandard sanitary facilities, or you have when we have a compromised hygiene practices, we share, toothbrushes, razors, etc., that can spread such kind of infection of the agents which can cause hepatitis. But it can also, some of the infectious agents pass through direct body fluid contact, such as blood, saliva, semen, etc. So we here are going to be concerned about, let me just put the point. We are concerned about this infectious causes of hepatitis, and especially those which are bloodborne. And if you recall that blood transfusion these days is a fairly safe method, and there are a lot of screens that are performed on blood that requires to be transfused and before a person receives the blood. However, although it was a huge boon, especially in the time of the Second World War, and there was a huge advancement in transfusion medicine, which increased the use of blood and various kind of blood component to be transfused. But it was also observed that while it saved lives, at a much later stage, majority of these recipients, they started developing inflamed liver and a condition which we just called as hepatitis. And that tremendously affected life as well as caused huge morbidity and mortality. So this event, during this time, it was Bloomberg who found that one of the reasons for this bloodborne hepatitis is hepatitis B virus. So he was also recognized with this prize. He was given Nobel Prize in Physiology or Medicine in 1976. And this discovery was a huge boon to this transfusion medicine, because they started screening for this hepatitis B virus and could definitely prevent a number of cases of bloodborne hepatitis. However, what was concerning was that bloodborne hepatitis continued despite HBV screening. During this time, there was another hepatitis virus which was recognized, hepatitis A virus, for which the screening was also available. Hepatitis A virus is not, it does not spread through body fluid contact, but it spreads through fecal oral route. But nonetheless, it also causes hepatitis, and it's also virus, and there were methods available at that time for screening both HBV as well as HAV. But of 100 cases, let us say, even after screening, almost 60 to 70% still developed hepatitis, and that was becoming a major concern. And people were worried about what is this unknown infectious agents. At this stage, they didn't even know whether it's infectious or not, or is it a variant of infection. And as we understand that if we want to make any kind of intervention, it is very important to identify the causative agent, which is the first step towards developing intervention strategies. So huge lives were lost, although blood transfusions were being made to save lives. One step when HBV was discovered and blood could be screened for HBV, but that did not take care of the entire problem, because despite such kind of screening, a large number of people who are getting blood transfusion continue to have hepatitis. They continue to die of hepatitis because of this blood transfusion. Or there was chronic cirrhosis and huge, compromised in the liver quality, and therefore the life quality as well. So here, Alter and his colleagues, they showed for the first time the presence of an unknown agent present in these undefined, cases of hepatitis, blood-borne hepatitis. So what they tried to, what they could demonstrate very systematically is that a large number of cases which continued to get hepatitis despite of HBV and HAV screening is because of a very unknown agent, and that possibly is a concern. So they call this mysterious illness causing agent as a non-A, non-B hepatitis virus. Non-B, sorry, not these cases, they called them as a non-A, non-B hepatitis because it didn't resemble either either HBV cases or HAV AV virus cases. So neither hepatitis A virus infection, neither hepatitis B virus infection. So what did they systematically try to do is that they took a set of people who had received blood transfusion, around 108 or so, and this appeared in the Lancet, and they observed that 12 of them had developed hepatitis. And this is despite the blood had been screened for hepatitis B surface antigen through a method called counter electrophoresis, and that is prior to transfusion. What they found is, out of these 12, 4 of them still had hepatitis B virus infection, and it was hepatitis B which was the reason for their illness. So this is even after screening for hepatitis B, and that was a concern. That would mean that the method that they are using for screening possibly is not robust enough, and some of them are missed. So they used a more sensitive method for testing once again these 12 patients, and they found, and they used the method radioimmunoassay instead of only using counter electrophoresis assay. Both of them are antigen antibody based assay, and they causes because radioimmunoassay has radio label, it is more sensitive. And what they found is that out of 4, 3 of the hepatitis case could be actually identified with RIA, and it was found RIA positive, but it was CEP negative in the earlier screen. So this still made 9 of them remain unidentified, and with this very systematic study, the team, or by Harvey Alter and his colleagues, they concluded that had RIA positive donors been excluded, 8 of the 9 residual hepatitis cases, that is almost 89%, would represent non-A, non-B hepatitis. The existence of previously unrecognized human hepatitis virus or viruses and is probable. They said it's probable because the reason or the agent was still unknown. And they continued their investigation. And so it was first Alter who recognized that there exists some agent which is neither A nor B and yet can cause hepatitis. The next question that they asked was that, is it infectious? Is it transmissible? So it was possible that even if it is hepatitis, if some agent is causing hepatitis in blood-borne hepatitis, it may not be transmissible. Is it infectious? So that was still a question. To address that, what Alter and his colleague did is that they took some plasma from some patients who had acute or chronic non-A, non-B post-transfusion hepatitis. And they also took some donors, but nonetheless, what you should remember is that they took some patients who were non-A, non-B hepatitis patient. They took their serum, and then they injected them in a model, in a chimpanzee model, and then monitored the chimpanzee model. You require to remember that chimpanzee is a different host, so it was not very, so it was possible that may not have the similar symptoms. But nonetheless, these chimpanzees first were screened so that they ensured that they are hepatitis A and hepatitis B negative, and after that, they were the serum from these patients were injected into a number of chimpanzees who were as a model animal. They followed by that, they performed various kind of biochemical, various kind of histological, assays to monitor the changes in the liver profile of these chimpanzees. And what they found is they found the kind of symptoms that they show, the kind of histology that they show of the liver was very similar to the hepatitis that they had seen in the patient, although there were subtle differences, like, for example, the incubation period in chimpanzee and possibly human were different, but those were expected because they are different hosts altogether. But nonetheless, what they finally could conclude is that this non-A, non-B hepatitis, the serum from these patients seems to be carrying a transmissible agent which can persist and which can remain infectious for a long time and which can cause an infection. So indeed, the agent is a transmissible agent. So with these kind of observations in the field, it generated a massive interest and a massive hunt for the transmissible agent, and it excited the whole research world, and a number of work where number of research labs started looking for this particular agent using very classical tools, many of the microscopy classical tools, classical biochemistry, centrifugation processes, various kind of biochemical molecular tools were applied to isolate, to see what is the agent in the non-A, non-B serum that is actually transmissible and is the infectious agent. Coming from one of the labs from India itself, from NIV, they studied non-A, non-B hepatitis patient, and they visualized virus-like particles in stool by immune electrode microscopy. Then people tried to really isolate them. So various kind of methods were developed, various kind of methodology were standardized for isolation of this virus and try to see and characterize this virus in their own way. So many of them said that, okay, can be the buoyancy of these virus, what can be the size of these virus, and many said that, okay, this is kind of seems to be between 30 to 60 nanometer size, and therefore it may not be a retrovirus. This is a very small enveloped virus is what it seems. So all this excitement was going going on. Once Walter and his colleague identified that there is indeed a transmissible agent in non-A, non-B hepatitis patients. So now the identification of this virus was the primary aim, and it was the highest priority. And although so much of hunt had gone, many years passed, and people could not get clue. Almost 10 years had gone in between, when it was Michael Houghton and his colleague. He was Chu Kyo, Weiner, Bradley, and Overby. All of them, this colleague, they were working in the pharmaceutical firm Chiron at that time, and that time they isolated a cDNA clone derived from blood-borne non-A, non-B viral hepatitis genome. So let us see what did they do, or what, especially for those people who may not have biology background, let us see what did they try to do. Now, why did it take so long for people to really not identify this virus, even though they were trying to, isolate it from the infected blood? They did not get sufficient titers. They did not get sufficient amount of RNA. So many people thought maybe it is a virus which does not replicate very well, which actually was not true. The turnover rate of this virus, which we later come to know, is fairly high. So they took this huge, difficult, laborious work, and this Michael Houghton's group, they started working. So what they did is they once again followed that method. They took some patient's plasma, and they infected certain chimpanzees, the liver, and then they took the liver cells and the plasma. They especially took those chimpanzees, tissue from those chimpanzees whose plasma contained very high titers of virus. That would mean there is a chance that they may get a lot of virus content. But the challenge was that whenever they were trying to take the tissue from chimpanzee, the chances are there will be huge amount of nucleic acid, the host nucleic acid, host DNA, RNA, rather than the viral genome. So nonetheless, they collected them, they subjected them to extensive ultracentrifugation to pellet the small virus, because a lot of information about the buoyancy of the virus, the size of the virus approximately was available at that time. So they recovered the nucleic acid, then they denatured the nucleic acid, and they made cDNA from both the RNA and DNA. They used certain random primers to make the cDNA from there. They made a library, and they made a library in a phage, and did all the molecular cloning and everything, and generated a huge library. And must appreciate their patients, because they screened almost around 10 to the power 6 recombinant phages and found one positive clone. And this one positive clone had a cDNA which did not match the DNA of host. So that was the amount, that was the systematic screening that was performed by Houghton and his colleagues. And eventually, they took this particular cDNA, they put it in another construct, so that from the DNA, a small peptide was made, and this peptide was expressed, and they called this fragment PS5. Now they had a protein. Now the assumption is not a protein, they had a peptide. So the assumption is that if this protein or peptide is a part of the virus, then the patients will develop antibody against it. And this is exactly what they tested. So what they did is an immunoblot. Shown here is the immunoblot. They put this peptide on several blots, and they used various kind of sera. This A strip is a patient sera, that means a human patient sera, and these are from the infected chimpanzee, the sera of infected, or plasma or sera of infected chimpanzee. So this set of chimpanzees were infected with non-A, non-B hepatitis virus. This set of chimpanzees were infected with HBV virus, and this set of chimpanzees were infected with HAV virus. This were used as a control to see that should not be one of those HBV or HAV proteins, and then one can say that it is not actually a different virus altogether. But what they found very encouraging is those the peptide could be recognized only in the serum or sera of patients who had NANVH hepatitis. And this was a remarkable thing, because what conclusion that came out of data is the identity of this stealth transmissible agent. So they concluded that this data indicated that this particular clone is derived from the genome of the blood-borne NANVH virus, and they termed it as hepatitis C virus, which was found to be belonging to the flavivirus family. And then a lot of work about the virus went on. But in the same issue of Science, there was a back-to-back paper. They took advantage of this, that this particular peptide can be recognized by the antibodies that are present in patient sera, and actually they developed an assay for circulating antibodies to major etiological virus. They called it major because even now they were not very sure that is it the only etiological virus for human non-A non-B hepatitis, because that part was still missing. But nonetheless, they used this particular clone, this continuous ORF. It was expressed as a fusion polypeptide with yet another protein, which was expressed in yeast, and they expressed it, they purified it, and then used it as an antigen in order to screen the serum from patients, or sera from patients of non-A non-B hepatitis. And that was a serological assay which was established, and this was a very major contribution, because this is the one which people were fearing of missing these this blood, who were tainted with some other kind of virus, which they were not able to screen because there was no assay established for that. So now they have a serological assay which tested sera from NANVH patients and they did it for they took serum from different parts of the world, especially from Italy and Japan. They took the serum patients from United States, Italy, and Japan, and they found that in all the patients of NANVH, that is non-A, non-B hepatitis patients, from all these different countries that they had collected, they all had this, they tested positive for this particular peptide. And that actually was a tremendous boost to the field, because this data suggested that HCV indeed is a major cause of chronic NANVH, that is non-A, non-B hepatitis cases throughout the world. And more importantly, this advent of this very specific sensitive test for HCV antibody that they described here can be used, can be further developed, or could be further developed to improve the safety of the world's blood supply, as well as provide a very important clinical diagnostic tool. So I think this was the contribution that Putin's group made to the discovery of hepatitis, the hepatitis C virus. So Alter recognized that there is a stealth agent which is infectious, which is the cause of this unexplained blood-borne hepatitis despite screening of hepatitis B virus. And Houghton identified, Michael Houghton and his colleagues, they identified this virus and called it hepatitis C virus. And not only that, they also could develop an assay that could be further developed or improved to make a screen for blood for screening various kind of blood for transfusion along with hepatitis B screening. So that was a tremendous step. But nonetheless, as here in the title itself, Michael Houghton's and his colleagues have written here that a major etiological virus with human. So one missing link remained, and that was, although it is nobody questioned that SCV is responsible for this undefined hepatitis cases after HBV screening, but question remained: is HCV alone responsible for hepatitis, or can there be any other reason and that is being missed? So if you all recall that it is important that to establish completely the that this particular pathogen is responsible for a disease, and if you go back to something which we know or understand as Cox postulates, it always said that you should be able to purify the agent, and once the purified agent is injected into an uninfected organism, the organism should develop similar symptoms and eventually should be able to again isolate the agent from the newly infected organism. So this link was still missing, and this link required it really required that the virus should be in pure form, and they required to have at least a clone or the genome of the virus, which should be infective and it can generate the virus which is HCV, and then it should cause the similar disease in any model animal that they are going to show. So this is where we identify with Charles M. Rice and his colleagues, and Rice and his colleagues, they worked very extensively on various the molecular various kind of, they applied various molecular biology tools in order to understand the entire genome of this new virus, hepatitis C virus. We can call it hepatitis C virus now. So, and eventually they actually identified a highly conserved sequence element at the end of 3' terminus of hepatitis C virus genome RNA. So what they once again did is they took the SCV RNA from the human serum and they used several synthetic oligonucleotides, they reverse transcribed it, they again made cDNA, they cloned it, and they did an extensive sequence analysis. And although several other studies had in part recognized various parts of the sequence of HCV genome, what they found is that the 3' non-translated region, it consisted of 4 elements, and amongst that, what was remarkable is that they found a novel sequence of 98 bases. Why this was a very important observation from their point of view is because that this particular results indicated that the HCV genome RNA terminates with a highly conserved RNA element, which is likely to be required for authentic HCV replication and recovery of infectious RNA from cDNA. So far, people had not been successful in making a clone or a cDNA clone or a infectious RNA, which when infected into a model or resulted into recovery of virus particles. So what Rice felt is that this may be the missing link, and they also started observing genetic variation in the isolated virus samples, and also try to figure out whether these kind of mutations that accumulate there are important for replication or they are restrictive for replication, and did a thorough analysis of the genome of your HCV virus. So having this as a background, they eventually could make a particular RNA variant of hepatitis C, and this definitely included, they gave the final proof, they engineered a completely fully functional clone of SCV, complementary DNA, and the RNA transcript from the clone were found to be infectious and to cause the diseases in chimpanzee. And when they had injected this RNA into the liver of chimpanzee, they found that it gave the characteristic chronic hepatitis symptom, and the virus was detected in the blood of the experimental animals. So this was the final proof. They could prove that it is hepatitis C virus alone that could explain this unexplained cases of transfusion mediated by hepatitis. So when they successfully, through genetic engineering, constructed or generated an RNA variant of hepatitis C virus, which included the region which the new region which they had identified earlier, and they injected it into chimpanzee, then followed the pathological changes in chimpanzee which resembled the chronic disease of human, and this kind of finally established that in that missing link, because the RNA was a pure RNA, it was injected into an organism which developed the similar symptoms, and the virus could be isolated from the infected organism. And this established this give the final proof that hepatitis C virus alone could cause the unexplained cause of transfusion mediated hepatitis. So this actually was the reason why Charles Rice was recognized for this Nobel Prize, and was a part of this Nobel Prize of discovery of hepatitis C. But with this with effort, there also began an effort to generate virus and cell culture based system to understand your various aspects of SCV biology. And eventually, what they did is helped by many other cells, many of the other labs who had identified a specific kind of clone. Especially this came from Japan, and many other groups, they made different kind of constructs, pseudoviruses, and this was mainly to help the the research community to have a cell culture based model of ex vivo virus generation. And they they could successfully make a complete replication of hepatitis C virus in cell culture. So they made several constructs and that could replicate in a cell culture of liver cells. They further improved on it, and not only the virus which was grown could be grown in the cell culture, they took that virus, they infected a chimpanzee, and they also infected a mouse model which had a human liver graft and could successfully infect as well as they isolated the virus from there and found that the virus can once again infect the cells that is the liver cell culture. So this establishing eventually a successful ex vivo culture of HCV. So all these things, and all because of these discoveries, what does it mean to us? It eventually meant that we could have a highly sensitive blood test which is available and which essentially eliminated post-transfusion hepatitis in many parts of the world, and it is documented almost that there's a decline, 80% decline in such incidents. Not only that, it also developed a tissue culture system effective for growing infectious virus that helped a lot of basic research and development of intervention strategies. And today we see that HCV is curable, and there are very specific drugs targeting specific points of HCV life cycle, where the assembly is targeted, the RNA dependent RNA polymerase is targeted. At the same time, the proteases can be targeted, and hepatitis C virus is curable today. And not only, the blood transfusion has become much safer. And it has also brought back the attention to transmissible hepatitis, which remains fairly a global threat to human health, and brings our attention once again to the various forms of hepatitis A, B, C, D, E, and the number of deaths that it still causes. Because it is preventable, cannot be accepted by the scientific community, and people should continue to work on these transmissible hepatitis. One point that is not known is that we still do not have a vaccine for hepatitis C. So this, there are open questions that remain in the area is that we still do not have a vaccine, and that is because of the heterogeneity, because of the high error rate of RNA-dependent RNA replication. At the same time, we do not understand the possibly required to have more understanding of HCV host factors influencing the disease outcome. We should be able to predict the hepatic fibrosis and also study more on HCV comorbidities. With this, I summarize once again that why the Nobel Prize in Medicine and Physiology 2020 was given to these three. So Harvey Alter identified there's a possibility of an infectious agent which can explain the unidentified hepatitis case, which were after HBV screening. Michael Hutton and colleagues, they identified the stealth virus and called it HCV. And finally, it was Charles M. Rice and colleagues who proved by making a construct, generating and engineering an RNA, which was when injected into a chimpanzee, could generate the same symptoms as chronic human hepatitis, finalizing that HCV, saving the world from this infection, helping the development of screens and eventually intervention strategy. So thank you, and sorry if I've exceeded the time. I'll be happy to take questions.
That was a wonderful talk, ma'am. Very simple to understand too. I would request the audience from the Gmeet, if they have any questions, they can unmute themselves and ask. No questions? Okay, then I would request the questions from the YouTube live stream be screen shared now. Yeah, thank you. Okay, so the first question is Asta Purvar asks, ma'am, working with mice is easier when compared to chimpanzees. So why did the researchers use chimpanzees as the model organism instead of mice?
So the mice is actually not a very good model for this particular infection. So not that any infection will mimic similar kind of symptoms or give similar kind of outcome in every model. So what with the experience of the other hepatitis viruses had shown them that it is not a very good, mice is not a good model, and chimpanzees, they showed a very good correlation with human hepatitis, and that's why they used. And if you recall the last slide on on the model of mice that they used was liver, which is from the human, was grafted onto the mice to make sure that the kind of observations that you would make will mimic that in human. So that is the reason why they went for chimpanzees, and possibly the model organism mice, which is, some of it is used today, are chimeric in nature.
Yeah, okay, ma'am. So due to time constraints, we'll have to end the session over here.
Okay.
The remaining questions, if possible, will be addressed at the end of today's session. So I apologize for the inconvenience.
I apologize for taking so much of time. Okay, thank you.
Okay, so I would like to thank Sharmishta ma'am to take time out of her schedule to present this talk, and I would like to thank the audience also for their participation. So now we conclude this talk. So audience, please stay tuned for the next talk, which will begin shortly. Thank you, everyone.
Hello, am I audible?
Yes,
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 Shiva,
please.
you're audible.
Yeah, shall I start now?
Yes, I think we can start.
Hi, Atreya, can I join?
Of course, sir. So
So
let me give
should I
let me my
share the screen? Okay, just a minute. Yeah, just a minute. Can you see?
yes, sir.
Yes, sir, it's visible.
Yeah. So
Yes,
I'll
sir.
just put it on a full screen mode so that it's easier for all of you. Is it okay now?
Yes, sir.
Yes, sir. Sir, you can hide the pop-up.
I can hide the what?
There's a small, yeah.
Okay, okay. Yeah, okay. So
Yeah.
let me know when should I start.
So I will give my intro speech and then you can start.
Okay.
Okay. Hello and good evening to everyone. That was an excellent talk by Professor Mr. Banerjee. Now we shall move on to the next and final talk of Nobelium 2020 webinar series with that in economics. We have here Professor Krishnendu Ghosh who will talk on the Nobel Prize in Economics 2020. Let me give a brief introduction about our speaker and to this year's prize winners. Before that, if anyone has any doubt or question during or after the talk, please feel free to post them in the chat box. We will read them to the professor for you. And people in the Google Meet, please keep your mics turned off unless asked to do so. Krishnendu Ghosh received his MPhil in economics in 1990 under the supervision of Professor Ramprasad Gupta, and later PhD in economics in 1993 under the supervision of Professor Anjan Mukherjee, both from Jawaharlal Nehru University, New Delhi. He pursued his postdoctoral research at Harvard University between 1997 to 98 with Ford Foundation postdoctoral fellowship under the supervision of Professor Eric Maskin. He currently is a professor of economics at Center for Economic Studies and Planning, Jawaharlal Nehru University, and has a teaching experience of more than 25 years. He had been a visiting research scholar at the Institute of Social and Economic Research, so Osaka University, Japan, during April to July 2010 and during the academic year 2013 to 14. His areas of specialization are oligopoly theory, auction theory, and game theory. He has published in leading economics journals such as Journal of Economic Theory and European Economic Review. He is on the editorial board of International Journal of Economic Theory and Arthnity Journal of Economic Theory and Practice. The Nobel Prize in Economics 2020, otherwise known as the Sveriges Riksbank Prize in Economic Sciences in memory of Alfred Nobel, has been awarded to American economists Paul Milgrom and Robert B. Wilson, both of whom teach at Stanford University, USA, for improvements to auction theory and invention of new auction formats. Having said that, Professor Dosidar, I welcome you to take over the virtual stage and begin your talk now. Sir?
Okay, thank you so much. And I must thank all of you for providing me with this wonderful opportunity. And I must also thank Professor Naresh Sharma of the Economics Department for connecting me to my young student friends. Okay, I have many slides today, but I'll try to be quick. Okay, so this year, my talk is on the Nobel Prize in Economics. So contributions of Paul Milgrom and Robert Wilson. By the way, I don't know whether you know this. Uh, Milgrom was a PhD student of Robert Wilson. So they are teacher and student. Okay. Now this year's Nobel Prize went to them. So the basic question that I want to ask is, what is it all about and why should we at all care for it? Okay. Now, I understand that many of you are not from economics background. So what I'll do is, in the first half of the lecture, maybe 15 minutes or so, I will lay out the stage. So that is, I'll speak about the basics of auction theory, and then, because in that context, discuss the contribution of these two persons. They're outstanding minds, no question on that. So let me start with, like it's begin with the beginning. So why at all do you need an auction? Now, just let me start with a story. Suppose you are lucky enough to inherit a Picasso painting. Now, in economics, we are always imagining things that are never true. Just imagine that you have. And now, suppose you want to sell it. You are in dire straits. You are in financial distress. Now, how should you go about it? Now, if you know the potential buyers and their maximum willingness to pay, the answer is very, very simple. You just call the person whose willingness to pay is the highest and sell it to that person at a price equal to his value or at a price slightly less than its value. He will lap it up. But the problem arises because you, the seller, has only incomplete information about the buyer's valuation. You don't know their willingness to pay. And no buyer would be willing to reveal this, okay? His or her willingness to pay. So to get the best price, you need to organize an auction because you don't know who is going to pay what. So you have to organize something called an auction. Now that's a very intuitive way of introducing auctions. Now let me come to auctions have been practiced in real life for many years. There is a very nice book by Cassidy, which I have mentioned here. It's a very dated book written in 1967. I have read that. And there he mentions auctions in Babylon in 500 BC and during Roman Empire. So it's quite ancient, right? Even before the birth of Christ, it was practiced. Now, in fact, the Oxford English Dictionary included the term auctions in 1595. And auction houses like Sotheby's and Christie's were founded in 1744 and 1766. Now, all over the world, auctions account for an enormous volume of transactions. Just to give you an idea how important is it, every week UK Treasury auctions of billions of dollars of bills, notes, and bonds. These are called Treasury auctions. Now, government procurement expenditure, on an average in the OECD countries, constitute about 13% of GDP. I'm sure in India also it constitutes a large, significant fraction of our GDP. Government in many countries auction off rights to drill oil and other natural resources on state-owned properties. In our country, examples are the coal block auctions, the spectrum auctions, things like that. Private firms also indulge in auctions all the time. And in fact, in internet auctions like the sites eBay, they also conduct auctions and they earn billions of dollars. The point is auctions are practiced in real life and they're very important part of the economic landscape. Now, auction theory as a discipline, I would like to stress the contributions of Milgrom and Wilson, but it has to be put in a context. That's why I'm giving this historical overview. William Vickery, who actually won the Nobel Prize in 1996, was possibly one of the greatest founding fathers of modern auction theory. His 1961 paper is a landmark paper. That year, he shared the prize with somebody called Jim Merlis. Now, in that year, the prize was not given for auction theory, but it was given something called incomplete information things, economics, okay? But essentially, Vickery and to some extent Merlis worked on auction theory. And their contributions were rewarded with Nobel Prize. In fact, Vickery later also worked on it in 1976. He has a landmark paper which initiated the idea of something called simultaneous ascending auction, which I'm going to discuss later. Now, the full flowering of this theory came in the 70s and 80s. So, and the big names are Roger Myerson, Eric Maskin, Paul Milgrom, this year's laureate, Robert Wilson, this year's laureate, and of course, John Riley, Samuelson, and Weber. In fact, in 2007, Hurwitz, Maskin, and Roger Myerson won the Nobel Prize for something called mechanism design theory. Now, auctions are special kinds of mechanisms. And Myerson and Maskin has also contributions in auctions and in mechanism design, which is related to auctions. Okay. Now, what exactly is an auction? I mean, I know all of you have some idea, but there is a specific definition. Now, auction is, as I said, it's a bidding mechanism by which some goods are sold. It's a selling mechanism. Now, it is described by a set of rules that specify how a winner is to be determined and how much he has to pay. So, who wins and how much does he pay? Now, the rules may even restrict participation. For example, you may impose a reserve price, which means bids below such a reserve price are not allowed. So what's a typical auction scenario? There is a seller who has a single indivisible object like a painting, house, or anything, or a coal mine, anything. I mean, you can have several objects as to be sold also. There is no problem. Now, the seller does not know how much any buyer would be willing to pay for it. That is, the buyer's values or the maximum willingness to pay for it are unknown to the seller. So it's a world of incomplete information. So the auction is intended to produce the best sell price. Now, what is a standard auction is where the object is sold to the highest bidder. I mean, typically that's what is practiced all over the world. You can theoretically imagine a weird auction where you can give it to the guy with the third highest bid, but in real life, it's never practiced. So that's why it's called a standard auction. Now, the value for a bidder is the maximum amount of money he would be willing to pay for it. And this is his private information. In economics, the term private information is used. It means that the bidder knows his own value, others don't know. Okay. And this bidder, I as a bidder don't know others' value. Others don't know my value. Okay. So that's called a private information. So value or willingness to pay is private information. Now, there is something called procurement auction. In a procurement auction is where the auctioneer is a buyer instead of being a seller. The bidders are the sellers. So what happens is, suppose University of Hyderabad, your university, wants to buy computers. Typically what it does is, or any public sector undertaking in India does, it is asked for quotations from companies. So, and the company which quotes the lowest price gets the contract. So, subject to some technical requirements of quality and other specification. That's called a procurement auction. So, in a standard procurement auction, the lowest bidder wins. It's just the standard auction in reverse. And in India, almost all public sector enterprises buy huge sets of items using procurement auctions, and Indian Railways are a leading player in this. They spend crores, thousands of crores to buy things using procurement auctions. Now, all over the world, historically, 4 kinds of auctions have been practiced. I mean, since many of you are not from economics background, let me go over with such auctions. The first is called a first price seal bid auction. What is happening out here? There is a single indivisible item up for sale. So bidders put their bids in sealed envelopes. So while they are bidding, they don't know what they are bidding. And the seller at the end opens the envelopes and checks for the highest bid. So the highest bidder wins and pays a price equal to his bid. Now, first price auctions are used extensively in auctioning mineral rights. Our coal block auctions use some form of first price auction. Okay, they are also sometimes used in sale of artwork and real estate and in procurement. Typically, your university, my university, they buy from the lowest quoting company. That's also first price auction in reverse. Okay. Now, the point is it's used quite frequently. Now, there is something else also. There's something called a second price seal bid auction. It's also called a Vickrey auction because William Vickrey was the first one to analyze it properly. The rules of the game in this auction are similar to the first price auction. However, here the highest bidder wins and pays a price equal to the second highest bid. Recall that in a first price auction, the highest bidder wins and pays the price he quotes. Here, he doesn't pay that price. I pay the second highest bid. That's why it's called a second price seal bid auction. Now, on the auctions website eBay, many auctions that take place are of this form. And in the US, in the stamp collecting business, second price auction is quite common. Although I must stress here, it's not as common as first price auctions. So in a first price auction, highest bidder wins, pays the price, whatever he has quoted. In a second price auction, highest bidder wins but pays the price which is equal to the second highest bid. Now there is another auction called an English or an ascending auction. It originated in England, that's why it's called English, and ascending because price rises. See, this is an auction which you often observe in movies and other places, and auction houses like Christie's and Sotheby's almost always use this. Now, here the bids are oral. So what happens is the seller first starts with a price and then bidders proclaim successively higher bids until no bidder is willing to bid higher. Okay. And the bidder who submitted the final bid wins and pays a price equal to his bid. So it's called an ascending auction because price keeps on increasing and bidders slowly drop out. I mean, until only one bidder is left. Okay. And then he takes the cake. Now, it's one of the most common types of auctions used all over the world. Okay. And Christie's and Sotheby's have been practicing this auction format for centuries, ever since their existence. I mean, all those Salvador Dali's, Picasso's are sold off using English auction. And the last kind of auction is called a Dutch or a descending auction. It originated in Netherlands. It's called descending because the price goes down. What happens is in this auction, the seller starts with a high price. He says, okay, this is the price. Are you willing to buy it? Nobody puts up his hand and says that I'm willing to buy. He reduces the price and he continues reducing the price till such time one of the bidders puts up his hand and says, okay, I am willing to buy it. So things start at a very high price. It goes down till there is an unwilling buyer. Now, traditionally, in the wholesale markets of flowers in Netherlands, tulips, for example, are sold off using Dutch auctions. Fish in the wholesale market in Israel, timber in Canada is sold using this auction. And the last point which I've mentioned here, when Google, the internet giant, went public, that was back in 2004, it sold its shares using some variant of Dutch auction. The point is all these four auctions are used all over the world. Now let me come to the basic model of auction theory. In economics, we always have models, okay? And there are some assumptions, and then we try to find out theoretical result and check for the empirical validity. Okay, or experimental validity. Now, modeling auctions. Now, auctions are modeled as games of incomplete information, and the basic tool is game theory. I'm sure many of my economics students, they have done game theory. And typically the relevant equilibrium concept that is used in auction is something called a Bayesian-Nash equilibrium. Nash was one of the most famous game theorists. You must have heard about his name. There is a film on his life also called A Beautiful Mind. Now, let me come to the benchmark model of auction. It's the simplest model of auction. Now, when I talk, first I'll talk about the benchmark model and some results, and then talk about the generalization of benchmark model, wherein the contributions of Milgrom and Wilson will be clear. Okay. Now, so what's the benchmark model? The simplest model of auction is as follows. There is one indivisible item up for sales. Think of a painting, and there are some bidders, say 100 bidders and one seller. So bidder's maximum willingness to pay is a private information. So I know my value, I don't know your value. Seller also does not know others' value. All I know for modeling purposes that others' value lie in some interval with some distribution function. There is it's a mathematical model. I am not using the mathematics. Okay. So now, this simple model is based on some assumptions. In economics, any model is based on some assumptions. So what are the assumptions of this canonical model? The first, it's called private values. Now, what is a private value? Now, the bidder's willingness to pay depends only on his own tastes and preferences, and it does not depend on what others' tastes and preferences are. So my value or my maximum willingness to pay will not change even if I were to know your value. Okay, that's called private value. Now, the second and the third assumptions are independent types and symmetry. I mean, it's just a technical assumption. It means that I know my value, I don't know other players' values. It's assumed that all values are identically and independently distributed. Okay, it's a statistical thing. And lastly, we assume that the bidders and the sellers are risk neutral. In economics, risk neutrality is used in a specific way, but I am not going into the details since most of you are not from economics background. So these are the basic assumptions of the benchmark model. Now let me quickly go over with the results of the benchmark model. What does the benchmark model? Suppose there is an auction where such assumptions hold true. Then what do you expect to happen? Now in a second price auction, see the winner's payoff is his value minus the second highest bid, because he pays the second highest bid. Losers get zero. They don't get the object, they don't pay anything. Now, what Vickery showed, and it's a very important result, that at an equilibrium, regardless of what others are bidding, any bidder will just bid his value. So, which means truth telling is an equilibrium. That is, irrespective of what your rivals are quoting, you are always better off or never worse off by quoting a bid equal to your value. Okay. So what will happen in the end? The person with the highest value will win. And how much will he pay? The value of the second highest bidder, because each one is bidding their values. Okay. Now, what one can show that for private value auction, the second price auction is equivalent to English auction. Now, just in a minute, I'll explain it to you. What happens in an English auction is price is rising and bidders are dropping off. At what price would you drop off? When the price reaches your value. Okay, so it's like as if you are bidding your value. And who is the second last person to drop off? The person with the second highest value. And who remains? The person with the highest value. How much does it pay? The second highest value. So effectively, English auction is equivalent to the second price auction with private values. Now let me come to the first price auction. Now this is technically more complicated. So what is the winner's payoff? It's his value minus the bid he quotes. The reason is in a first price auction, the highest bidder wins and he quotes his and he pays whatever he has quoted. Losers don't get the object, they don't pay anything. But note that since the winner pays the amount he has quoted, it is never optimal, never in his best interest to bid one's value. Hence, in this auctions, players must bid below their values. But there is a slight catch. The lower you bid, the lower is your probability of winning. But it is also true that the lower your bid is, the higher is your payoff, conditional on winning. That if you win, then I'll get a higher payoff because I have to pay more. So in equilibrium, these two trade-offs must balance each other out. And there is a precise formula for this and at a theoretical equilibrium. So I have not written it there, and auction theorists have found that. Okay. Now one can again show worst price auction is equivalent to Dutch auction. See, what happens in a Dutch auction is that price is descending and you have to decide at what price you have to say, yes, I'm willing to buy. If somebody else buys it before you, you cannot do anything. So essentially this is strategically the same as a first price auction. So first price auction and Dutch auctions are just the same. They lead to the same equilibrium outcomes. Now, auction theory has a very important theorem called the revenue equivalence theorem. Now, Vickery first showed that the expected revenue or the price that you get in a first price auction is always equal to the expected revenue in a second price auction, in the benchmark model, that is, in the model where those assumptions are satisfied. See, in a first price auction, the highest bidder wins and pays whatever he has quoted. In a second price auction, the highest bidder wins but pays the second highest bid. But on an average, the expected sale price at which the good will be sold is the same across these two auctions. In fact, this theorem is very general. And Meyerson, another Nobel laureate who won the Nobel Prize in 2007, had the most general version, and he showed that in the benchmark model with independently and identically distributed values, the expected revenue from most auctions are the same. And you can have a whole lot of auctions. I mean, I'm not going to do that, and it's a very powerful theory. But just remember, it's only valid in the benchmark model with those specific assumptions. If you drop those assumptions, then revenue equivalence will not hold. But it's a very fundamental and benchmark result. Okay. Now, before coming to the generalization of the benchmark model and Milgram's contributions, let me talk about something called optimal auctions. Because suppose, think of a problem theoretically. You have something to sell. Now, what is the best way of selling it? You can use an auction, you can use some other mechanism like take it or a leave it offer, anything. Now, is there, at least in principle or theoretically, a best way of selling the object so that your expected price that you get is maximized? Now, Meyerson, in his seminal paper in 1981, solves this theoretical problem, and he shows how to derive optimal auction for a large class of problems. Now, this was a remarkable achievement, at least theoretically, but there was little connection to traditional price theory or traditional economics as we know it. But that gap was filled up by Bulow and Roberts in a very nice article in 1989, and it shows that Meyerson's analysis is essentially equivalent to standard monopoly third-degree price discrimination. Some of my economics student friends would be able to understand this. Now let me come to the generalization of the benchmark model. Recall that the benchmark model was based on some assumptions like private values, symmetry, independence, risk neutrality, things like that. Now, auction theory is rich enough to drop all those assumptions one by one, relax them one by one, and see what happens, theoretically as well as empirically. So one such thing is asymmetry. So distributions are no longer the same. Risk aversions, bidders are no longer risk neutral, risk averse. Last two are very important. Values are interdependent. That if I come to know of your value, I'll change mine. In a private value case, it's not true. The benchmark model is that of private value model. That even if I come to know your willingness to pay, I will not change mine. However, in interdependent value model, this is not true. Your value for a painting may depend not only on your private information, how much you like it, but also on somewhat on others' private information, how much they like it, because this affects the resale value and or the prestige of owning it. And in real life, this is very true. Then you can also think of relaxing the benchmark model by allowing for correlated types. That is, in the benchmark model, we assume that I know my value, I don't know others' values. I assume they're identically and independently distributed. But suppose there is correlation among other values. And also, lastly, there's something called multi-unit auction, where there is more than one unit is up for sale. The benchmark model is based on a single object auction. Now, Wilson and Milgrom has path-breaking contributions in these things, interdependent value auctions with correlated types and multi-unit auctions. And I'm coming to that. That's why they won the Nobel Prize. However, I must stress here, apart from auction theory, they have contributions in other areas also. Now, let me first talk about something called common value auctions. Common value auctions is a value of the object being sold is the same for all bidders, but this value is not known at the time of bidding. A typical example is bidding for rights of exploration of some natural resources, say oil. Okay, or coal. The coal block auctions are common value auctions. The amount of oil or coal available in a particular area is fixed, but it is really known after the auction and when the successful bidder starts trading. See, prior to the bidding, you have imperfect information about it. Each company has employs its own geologists. This gives some estimates, but nobody knows the real value. There's some estimate, but it's called common value because the value is the same, because the amount of oil or coal is the same for everyone. Now, Wilson, one of the laureates this year, showed us how to theoretically analyze such auctions. At that point of time, it was very challenging problem theoretically, but Wilson in his 1969 paper, and he extended that in 1977, provided us the way how to analyze such auctions. Now, it's the other end of the spectrum. In a private value auctions, the value, it's known to you and you wouldn't change that value even if you were to know of other people's willingness to pay. Common value is the other extreme, where the value is same for everybody. Only thing, I don't know what the value is. I only have imperfect signals about it. Now, there is something attached to common value auctions, and that's called a winner's curse. Now, what's a winner's curse, you may ask me? I mean, after all, winner is a winner. But each bidder, so it starts like that, must recognize that he wins the object only he has the highest signal. So if my signal is the highest in an equilibrium, I win the auction. However, the fact that he has won means that other bidders, his rivals, have lower signals. Now, this is sort of a bad news in the sense that if I must now take that into account, and my expected value conditional on winning will go down because other signals are lower. In the beginning, I don't know anything. If I win, I know mine is the highest signals, others are lower. So my expected value conditional on winning will go down. The failure to foresee this effect and take fully into account when formulating bidding strategies will result in what has been called winner's curse, the possibility that the winner pays more than value. Okay. It is possible in practice, and this is said to happen often in practice, and there are empirical studies of that companies have landed up paying more than the actual value of the object. This is called a winner's curse. Now, however, I must say winner's curse will not take place in a theoretical equilibrium. In equilibrium, bidders will take into account the possibility that they might overestimate the true value of the object and will bid more cautiously. So if you are a rational bidder, you should factor that in, that if I win, I have to then decrease the estimated value. So I should bid accordingly. So winner's curse cannot take place in an equilibrium, in the theoretical equilibrium. Now, Wilson provided the first theoretical analysis of this, and his two papers are beautiful. At that point, people didn't know what to do with it, how to analyze it. He showed us the way. So that is one major contribution of Bob Wilson. Now, just to give you some historical anecdote on winner's curse, I got this from an article by Shubik in this book, which is mentioned here. See, apparently in AD 193, the whole Roman Empire was sold by an ascending auction by the Praetorian Guards. The winner, and therefore the next emperor, was Didius Julianus, okay, who just reigned for 2 months before being overthrown and executed by Septimus Severus. He was actually beheaded, and it's said to be a very tragic case of an winner's curse, okay. Now, I got this anecdote from Shubik's article. Now, let me come to this interdependent value auction. I talked about generalization of the benchmark auction. So what's the benchmark auction is based on independent private value model. Now, independent private value and pure common value are limiting cases of more general information setting. The interdependent value auction is something in between, that my willingness to pay depends not only on my private information, but also your private information, which I don't know. Now, here Milgrom and Weber in these two classic papers laid the groundwork for the analysis of such auction, and they have an extremely important contribution, Milgrom especially. So what's the general model? A bidder's private information is summarized as the realization of a random variable, his signal. Earlier in the benchmark model, the signal was his value, his maximum willingness to pay. Now here, the value is a function of not only his signal, but also other bidders' signals, which he doesn't know. So this specification allows us to include both the private value model as well as the common value model. So that's the beauty of it. And Milgrom and Weber in their very landmark paper provides the complete analysis of this model where values are interdependent and signals are affiliated. Now, just one thing, affiliation is a strong form of correlation, which roughly means that if a subset of all signals are large, this makes more likely that the remaining signals are also large. By the way, auction theory is very mathematical, and it not only uses mathematics, but lots of statistical tools, okay? So the basic tool is game theory, which is of course a branch of mathematics, and then you need to be very, at least somewhat good with statistics also. Now, Milgrom in his paper, it's a landmark paper, shows that in this interdependent value model with affiliated signal, the following is the revenue ranking. What he showed that the expected revenue with English auction is greater than equal to the expected revenue with the second price auction, and it is greater than equal to the expected revenue with the first price auction. So no revenue equivalence here, unlike the benchmark model. Okay, now in this model, the English auction fetches the highest expected revenue, so it's best for the seller. No wonder that all over the world English auction is so common, because typically most goods have some interdependent values. Private values are an extreme case. common value is an extreme case, although it is observed. I mean, mineral rights auctions are common value auctions. Okay. So therein Milgram's contribution. Now, Milgram, let me also talk about something called a linkage principle, and it is related to this revenue ranking result. Now, Milgram showed that he has done insight that certain auction format generates higher expected revenue than another whenever its prices better aggregate the private information of bidders. Now, consider, for example, an English auction with seal bid second price auction. Now remember, recall that I told you with private values, these English auction and second price auctions are equivalent. Because what happens in an English auction is that with private values, when the price reaches your value, will drop off. Now here with interdependent values, it's different. Suppose you are a participant in an English auction. You observe somebody dropping off. From there, you get an inkling of his signal. You factor that in your value function and revise your value estimate. Okay, so you are able to get more information and aggregate it. And that therein the linkage principle comes into being, and the linkage principle assures that expected revenue is higher in English auction than in second price auction with interdependent values. It's with private values, it's the same. By the way, the Dutch auction and the first price auction continue to be equivalent, even with interdependent values. So this revenue ranking result is an extremely important result which Milgram gave us. And there is something called efficiency issues also. An auction allocates efficiently if the bidder with the highest value is awarded the object. Because you would always like it something. Say, suppose you are selling off a factory or something. You'd like it to go to somebody who values his most because he is likely to use it more properly, better way. Now, in the benchmark model with private values, equilibrium outcomes are always efficient. However, in the interdependent value, this need not be the case. The reason is the highest signal need not be the one with the bidder with the highest signal, need not be the one with the highest value. Okay, so even symmetric equilibrium can be inefficient, and then later people have found out under what conditions efficiency is achieved. A student, she found a result called a single crossing condition. If that is satisfied, then efficiency is guaranteed in an interdependent value model also. Now let me go over to the other thing, the last generalization, that since the 90s, the major research focus shifted from single object auctions to multi-object auctions. I mean, the shift was because a lot of things were multi-object, like spectrum frequency bands, electricity batches of troubled date, and the number and size of the transactions of these objects are huge, and even small efficiency gains in percentage terms can be worth billions. Now, multi-unit auctions or multi-object auctions, where several things are up for sale, spectrum auctions are an example of that. Now, they lead to enormous complexities. Now, auctions of a large number of interrelated objects raise issues that had not come up in the analysis of standard single unit auction form. One thing is complementarity, the other is externality. I'll just explain it with a very simple example. Suppose there is a spectrum A and spectrum B. A company values spectrum A at $10 million, spectrum B at $10 million separately. But if it gets both these together, it is valued together at say $40 million. It's not 10 plus 10, it grows up because there is some complementary externality between them. There are synergies. So if it gets both together, it's willing to pay more than 20. But for separately, if it gets just one, it's just willing to pay up to 10. So that's the idea. Okay. Now, multi-object auctions raises such kinds of issues. And so it called for invention of new auction formats. And in this respect, again, Milgrom and Wilson had both of them have path-breaking contributions. So what did they do? I'll quickly go over to them and end my talk. They formulated new auctions. What are they? I'll just explain. Simultaneous ascending auction, which Milgrom and Wilson developed for the United States Federal Communication Commission radio spectrum auction back in 1994. By the way, I must state, before 1994, mostly the spectrum prices were decided by government bureaucrats or politicians. Okay. However, for the first time, they actually invited theorists. Milgrom and Wilson were hardcore mathematical economic theorists to design auctions in such a way so as to maximize the revenue accruing to the government. And to do that, they designed something called simultaneous ascending auction. Now, other formats designed by them include share auctions, which Wilson did in 1979. Then there's something called a combinatorial clock auction. Again, Milgrom had a role in it, and incentive auctions. What I'll just quickly go over with go over all the auctions formats. But just to let you know, I'll first discuss share auctions in one slide, and after that, the other three. First, the simultaneous ascending auction, and then there are problems with it. So to circumvent that, combinatorial clock auctions were introduced. Then some drawbacks were found out, and to circumvent that, incentive auctions were introduced or designed. And Milgrom and Wilson had contributions, path-breaking contributions in all of them. So what's a share auctions? Wilson 1979 paper, again a remarkable paper, was much ahead of its time. So what's a share auction? Auctions in which each bidder offers a schedule specifying a price for each possible fraction of the item, say a certain volume of treasury notes. Now Wilson's one of the major results was that he showed that in a uniform price auction, when all the shares are sold at the same price that equates supply and demand of shares, there are Nash equilibria that look very collusive. That is, prices are not that high, as high as the seller would like to be. Okay, so support prices are much lower than if the item was sold as an indivisible unit. So now that was Wilson's, at least the theoretical contribution behind share auction. Now, what's a simultaneous ascending auction? It's a huge area. Just to tell you, it's something like English ascending auctions, but several units are for sale and it's simultaneously. Several spectrums. So spectrum rights for Delhi, Calcutta, Bombay, Hyderabad are up for sale, and companies are bidding. Unlike most auctions in which bidding is continuous, simultaneous ascending auction have discrete successive rounds with the length of each round announced in advance. Now, participants are allowed to bid on multiple rounds. And after each round closes, results are declared. Okay. Only then do bidders learn about other players' bids. And it continues till no such object is left unsold. Okay. Now there are of course other nuances. I'm not going into that because time is short. Now regarding applications, it has been applied all over the world, starting with the 1994 FCC spectrum auction. It raised $20 billion. Then in 2000, the United Kingdom, its 3G spectrum auctions raised $34 billion. Again, it was designed by two theorists, Binmore and Klemperer. Binmore is in fact a mathematician who works on game theory. Now, versions of this auction have been used in many countries, including our country. Our 3G auction was also a copy-paste version of that. We, being a copy-paste nation, of course, copied this. Okay, and this generated hundreds of billions of dollars for governments worldwide. However, I must say there has been some spectacular failures for even the simultaneous ascending auction.
Excuse me, sir.
Yes?
Excuse me, sir.
Yeah, tell me.
So we have only 2 minutes. Like, could you quickly repeat?
Yeah. I'll just. So simultaneous auctions had some problems. And drawbacks, I'm not noting, it's something called an exposure problem because it does not allow for package bit to circumvent that. Milgram designed something called combinatorial clock auction. Again, it was applied all over the world since 2008, but some drawbacks were found. To solve that, Milgram went for incentive auctions. Again, Milgram's contributions. But later it was found out this itself has some huge computational complexities. So the search for a perfect auction continues. And what makes an auction successful? There is no silver bullet. There are many things you have to consider. And lastly, why should we at all care to know some auction theory? Okay. Now, there are many reasons, and I just want to stress the last that read this book, Why Every Economist Must Know Some Auction Theory. And the main reason you should read auction theory because it's great fun. Okay, and these are some reading lists I've given at the end of the slides. And so thank you. And that's what I wanted to say.
Thank you, sir. That was an excellent talk and was easily to comprehend. I think it's now time for the question and answer part. If anyone has any questions, please ask from the Google Meet.
May I? May I go ahead?
Yes, ma'am.
Yeah, you can ask, please.
Completely from a different background, but was trying to really understand. So, how is it auction thing related to? They say the taxpayers. At what level the taxpayers apply this concept of auction? Something which is not clear.
The taxpayers.
Yes, they say that
Yeah,
this
yeah.
auction theory can
See,
also benefit
the
taxpayers
Oh,
in the
yeah,
sense.
yeah. See, the government, when suppose think of Indian government, when it sells of 3G spectrum auctions through the simultaneous ascending auction, it got huge amounts and that comes into the coffers of the government as a revenue. So that helps the taxpayers because the government uses that money, right? And
I was under the impression with the when you are paying a tax or something at that level, also auction theories can be applied. I was under that impression. So,
yeah, of course.
okay, thank you.
Anyone else? Okay, if not, like we can ask from the YouTube live stream. Shubham, they ask if the revenue generated in English auction is higher than the second and the first auctions, first price auctions, then what did they originate for and why were they even practiced if the auctioners had less profit from it?
Okay, okay. The answer to that is this is true only in the interdependent value model. Okay, if the values are interdependent. In the private value model, the revenue equivalence holds. So the English auctions revenue would be exactly equal to that from second price or first price. So that's the answer. So it depends on the context. It's not always higher. Okay.
Okay, Aditi asked, as you mentioned, if you want to sell something like a Picasso painting, you have to go through an auction. Does that mean the price of anything I Google has been decided by some kind of auction behind the scenes?
In principle, yes. Think of what is a price of something. How does one get into a price? I mean, always there is some kind of implicit auction theoretic thing behind it. When a seller sets a price, he sets it okay, possibly okay, I'll set it because I guess these are the willingness to pay, and I should set it at a price which will maximize my chances of selling it. Okay, something. So there is an implicit auction going behind any price discovery process. In fact, there are papers on it also.
Okay. How is the base price of the auction decided if it is certainly not the making price or the raw material price? Is the base price based on previous auction price?
It depends on the context. Sometimes it's based on the previous auction price, or Sometimes the seller may set it optimally. Okay, he has some idea of the values of the bidders. Okay, then at what reserve price should I set to maximize my expected gain? There is a whole literature on it. Such literature is called an optimal auction literature.
Okay. Then do you think that the economic divide in the world is largely because some goods and services get sold on a higher bid than others?
I don't know the answer to this. It's a very fundamental question. Economic divide in the world is too fundamental. I really don't want to venture out to answer this.
Would analysis of auction results depend on the number of bidders, even though it is not constant throughout?
Analysis of auction results, yes, it depends on the number of results. The revenue that you would get, expected revenue, depends on the number of bidders. Higher is the number of bidders, higher is the expected revenue that you would get. Because competition pulls up the bids. Bidders are forced to bid more. Certainly, yes.
Yeah, could you screw it up? Like sixth question.
Sixth question is, what are the inefficiencies in auctions process of auctioning? Now, as I said that in this auction theories, an outcome is said to be inefficient if the object lands up in the somebody whose value is not the highest. And we saw that interdependent value auction, this may well be the case, even in theory. And in practice, it often happens. In fact, there is an entire literature called efficient auction, how to design auctions in such a way so that the object lands up in the hand of somebody who values it most, to ensure efficient outcomes. That is.
Okay, I think we have come to the conclusion part. So with that, once again, sir, we thank you very much for accepting our invitation and providing that extraordinary talk.
Okay, thank you so much, all of you. I wish I could meet you someday.
I hope this session was useful for everyone. I am now signing off by asking Aditi to give the vote of thanks. Thank you. Thank
Okay.
you.
Thank you, Nanda. Before the vote of thanks, I would like to address Aditya to ask questions to Sharmishta ma'am from our audience.
Yeah, we shall have a change of pace now and move into the field of medicine again. Okay, so the first question was asked before the economics talk session. So the second question is from Ananya. What do you think is the most distinguishing feature to the discovery of this virus slash pathogen that led to its recognition for a Nobel Prize?
I think the simple answer to it is the number of lives it saved from hepatitis from an unknown blood-borne cause, which went undetected. Almost, if you think about, even now we say 3% of the world's population are infected with HPV. Even now, chronically, we say that. And imagine when it was not screened, and imagine when it was not known, and just imagine when the person gets a transfusion thinking that all is safe with the blood and gets an infection because of this unknown cause. And because majority of the blood-borne hepatitis was not because of HBV, it was eventually found to be SCV, which was the culprit, and that was the basis, the life-saving, discovery and discovery, recognition of the virus, eventually leading to a screening method and a lot of molecular biology tools that eventually led to intervention strategies in the form of medicines, I think, is the main essence of giving these three this much of recognition, that not that it demeans the other things, but I think, yes, it's a very important contribution.
Okay, ma'am. Next question. Aditi asks, can you elaborate on autoimmune hepatitis?
Okay, so anything that is autoimmune is generally a condition when the antibodies are actually generated uh, against some other non-self molecule, and eventually because of cross reactivity, or it tends to recognize a molecule of your own body. And when such a situation happens, these antibodies start attacking the body's own cells and leading to an autoimmune condition. So the tolerance to self is lost. And in case of autoimmune hepatitis, many a times it can be because of certain infections, certain other kind of pathological conditions. The antibodies that are circulating in the body may attack liver cells, causing its damage. And that's the situation that can lead to an autoimmune inflamed condition and leading to hepatitis-like symptoms.
Okay, ma'am. The next question. Ashutosh asks, what are the differences between hepatitis A, B, C, and D?
Okay, so uh, I mean, you want to know about the symptom wise, there are various, so A, B, C, and D. So there is also E, if you recall. So hepatitis A is primarily, definitely the nature of the virus. Okay, all of them belong to different family of viruses. But at the same time, their mode of infection also is very different. B and C are actually the bloodborne, whereas A has a transmission through fecal oral route. So contaminated food, water, and unhygienic practices, not washing hands. These can actually, so you come in contact with an infected material, like not safe drinking water or contaminated food. So that can actually lead to infection by hepatitis A virus, and it causes acute hepatitis, and there is no as such medicine for it because it is self-limiting, and the patient is cured after some time. But bloodborne hepatitis B and hepatitis C can be very severe, and they can become chronic, leading to cirrhosis and also liver cancer. So B and C then transmits through body fluid contamination. D is kind of dependent on B, and hepatitis D virus requires the envelope protein of B, and therefore generally occurs with hepatitis B and D together. So all of them belong to completely different family of viruses. You can look. I had a table for that, which I had to rush because of lack of time. But A and C are RNA viruses, and I think D has a circular RNA, and B is I think it's a DNA virus. So with uh, so these are some of the differences, and if you are looking for more specific differences, possibly that can be elaborated. But the mode of transmission and the nature of the virus that causes it are the principal differences. The symptoms are much milder for hepatitis A, and but it can be very severe for hepatitis B and C.
Okay, ma'am. The next question. Aditya asks, is the mode of action slightly different in the different hepatitis viruses, which created a difference in their detections?
Mode of action, you mean to say the mode of replication and all? Yes, it does. It is different in different viruses, but so if you are trying to say that if that is the reason which created differences in their detection, um, I mean, I'm trying to understand difference in the detection during the time of discovery, or are you trying to say that how they are diagnosed? If somebody can make it a little bit more Clear to
Ma'am,
me?
like detection during like the confirmation or the discovery time.
Or that in the post process of discovery? No, so I think it's not the mode of action alone. I believe that it is also the titers, the ease of getting the genetic material, or a predominant your antigen, that would make a difference in how you can quickly isolate the virus for the purpose of identification. So mode of action definitely is different for all the viruses, and that eventually what you call it translates into differences in the titers, or differences in the antigenic expression of the antigenic viral protein, etc. So indirectly you can say that, but I think it will also depend upon the kind of technology or kind of approach that you are using to in order to identify them.
Okay, ma'am. The next question, Manaswita asks, do transfusions prove effective to cure hepatitis C virus?
Transfusions. You mean to say blood transfusions?
I believe, yeah, that blood
So,
transfusion.
so
Yes.
the hepatitis
Blood.
C virus from the infection comes from a tainted blood transfusion, and the hepatitis C virus can be cured by a proper. There's a treatment regimen for around I think 6 months or so. There's a combination of direct and direct viral drugs are given, along with sometime interferon therapy is given, and that is the one which is actually effectively cures hepatitis C. And yes, it is fairly curable. You can completely see loss of viremia, loss of viral titers from the periphery, and that is considered as cure. But we do not have a preventive vaccine yet. Uh, mm, so we don't do blood transfusions to cure hepatitis C virus, but there is a proper treatment available for it.
Okay, ma'am. The next question, Vidya asks, why is it that hepatitis C only spreads through blood? Any particular reason?
So every infection has a route of entry, and for example, hepatitis A has a fecal oral route because it enters through that, and then it interacts with mucosa, gut mucosa, and possibly gets the entry. So it depends upon from there how uh, every pathogen will have a specific route of entry, and hepatitis C virus has this blood-borne route. Hepatitis B also has a blood directly body fluid route, maybe because it cannot survive the other route of entries, so it tends to get disseminated, and it cannot survive the other route of entry. But a direct contact between fluid to fluid of infected, it is taken into bloodstream, and then through portal circulation it reaches liver. That is what the best I think I know about it.
Okay, ma'am. I think those are all the questions. Thank you so much, Sharmishta ma'am, for answering them.
Thank you, and thank you. Sorry for rushing towards the end.
Yeah, okay, so with this, we have come to the end of the Nobelium 2020 webinar series. I now ask Aditi to deliver the vote of thanks.
Thank you, Aditya. To respected faculty coordinators, speakers, and my dear audience present, it's my privilege to have been asked to propose a vote of thanks for our first ever event as JSC coordinators. On behalf of Junior Science Club and Entropy, I would like to extend a hearty vote of thanks to all the honorable speakers who shared their virtual presence and took out valuable time from their busy schedules to share their knowledge with us. I must mention our deep sense of appreciation for Dr. M. N. Rajesh and Professor Soumya Dechamma, who gave the first two talks of the 3-day webinar series on World Food Program's Effort to Combat Hunger, and the poetic voice of Louis Gluck, respectively. I also would like to extend my thanks to Dr. Soma Sanyal for her talk on general theory of relativity, Dr. P. Chandrasekhar for his talk on CRISPR, Professor Sharmishta Banerjee for her talk on discovery of hepatitis C virus, and Professor Krishnendu Ghosh Dasidar for his talk on improvements in auction theory and inventions of new auction formats. I must tell that I thoroughly enjoyed this talk and understood most of it being from a non economics background. Your talk really gave me a large perspective of auctions from what I previously knew from movies and TV shows. Apart from all the speakers, I would like to express my gratitude to the Dean of the School of Life Sciences, Professor Dayanand Siddhavatam, Director of College of Integrated Studies, Professor Sanjay Subodh, and the Associate Director of College of Integrated Studies, Professor Krishnaveni Mishra, for providing us with this opportunity and support to organize webinar series during the ongoing pandemic of COVID-19. I'm very grateful to all the faculty coordinators of JSC and Entropy, Professor S. Ilangovan, Professor Sharmishta Banerjee, and Professor Srinath for their constant presence, guidance, and valuable encouragement. At last, as no program can be successful with a single person, so I extend my big thanks to the CIS office members, our senior and fellow student coordinators of Junior Science Club and Entropy, and our dear audience for participating in the webinar. Thank you so much for making this event successful with your contribution. And now I would request all of you to please follow us on the social media handles. And I hope that the feedback form will reach you via email very soon. So please put in efforts and some valuable time of yours to give us a feedback on this event. Thank you so much.
Thank you all.
Thank you, ma'am.
Yeah, thank you so much. Thank you so much, Aditi.
Thank you, sir, and thank you, ma'am. Thank you a lot
Okay.
for your talks.
Okay, take care from Corona, all of you. I'll leave now.
Thank you so much, sir.
Thanks.